Good morning, everyone. First, I'd like to thank the organizer giving me the opportunity to present our work today. It was a great honor to share a podium with Ali and other excellent immunologists to develop the XCV vaccine. Today, I'm going to just share a journey that we took to develop a XCV vaccine. I'd like to also acknowledge Ali because she did a fantastic introductions. I don't have to tell you why we need how important the XCV vaccine is needed. And I like to echo Ali too, is that this is a story that we try to conquer efforts. It's one of the routes that we take. By no means is the only routes that can take. The story is a developing story. Unfortunately, we don't have a finished product yet, but we are very confident and we know kind of what type of immune response we want from our vaccines in order to confer XCV protections. But more importantly, with Ali saying is that with the GSK vaccine failure recently, we really need more candidates to be tested in order to explore a variety of different ways to induce immunity against XCV infections. So here's my disclosure. So two vaccine has made to phase 1 clinical trial. The other one is so-called T-cell vaccine. And the other one that we are working on is a subunit-based vaccine. What we're focusing on is using the envelope proteins on the surface of the virion to induce immunity. We believe that we take good care of maintaining the native structures of the glycoproteins. We believe that by doing that, the antibody that targeted this subunit will similar to antibodies that target the actual virion. And this vaccine will be much more safer because they don't need to productive infections. The drawback of this is that making this glycoproteins in a native form is hard to do. We make them in the CHO cells. So far is the only XCV vaccine can show efficacy to protect chimpanzee, the only immunocompetent models that we use. And unfortunately, also the chimpanzee model is no longer available. The vaccine do not prevent acute infections and can reduce chronicity of XCV infections. This is an important concept for the vaccinology for XCV because we know that the pathologies of acute XCV infection is relatively mild. And most of the disease is manifested at the chronic states of infection. So if we can actually reduce the chronicity of XCV vaccine, I think that's can be considered as a successful strategy. So this data is just to summarize some of the earlier work that Mike has done in Chirons, looking at the vaccine efficacy in chimpanzee. As you can see, after animal that either vaccinated with the glycoproteins vaccine, only about 15% 15 to 20% of the animals develop chronicity, whereas up to 60% of the non-infected, non-vaccinated animals develop chronicity, suggesting that vaccinations can reduce XCV chronic infections. But like Ali highlighted, the diversity of XCV is one of the big obstacles for vaccinology. On the right is the phylogenetic tree of different XCV vaccines, of different XCV viruses. As you can see that the diversity is much higher compared to even HIV or HPV. So meaning that, like from our previous slide, our vaccine can induce immunity against genotype 1 virus. Can it actually also protect other genotype virus of XCV? This is an important question to answer. So we take the similar sera from these vaccinated chimpanzee. We use a models in the tissue culture to test whether these sera can protect XCV entry. So as you can see on your left-hand side, you can see the vaccinated sera can protect entry of XCV particles from genotype 1A and also confer protections against entry of of XCV particle against other genotypes that represent a major clade of the XCV particle, albeit is that genotype 2 and 3 seems to be a little bit reduced. But overall, the anti-sera can protect at some level to prevent entry of the XCV particle. And the most important question is that can this also happen in humans? And so after the phase 1 clinical trial testing the vaccinations of the glycoprotein-based vaccine, we take these sera from the volunteer, we test their ability to block XCV infections in the tissue culture model that we have. These are 3 volunteers that we pick, and then we test their cross-neutralizations activity across all 7 major genotypes of XCV particles. You can see that there's each individual seems to have a variety of different effectiveness in terms of blocking the XCV entry, but nonetheless, I think they show a cross-protective response to against many different genotypes of XCV particle, giving us a confidence that our vaccine can induce antibody, and these antibody can neutralize many different clades of XCV. So what does that mean? So this is not data from our lab, but it's highlight the important role of neutralizing antibody in preventions of XCV infections. So these mouse model basically harvests human liver, so they can be susceptible for XCV infections. What they're testing here is that before they infect these mice with XCV virus, they either incubate them with control antibody or antibody that's isolated from chronically infected patients. These chronically infected patients have a lot of neutralizing antibody against XCV in them. So we're testing whether these neutralizing antibody can protect XCV infections. On the left, you can see that in the blue square, basically is that the virus can quite effectively replicate in this animal model. But on the other hand, additions of the chronic sera from these chronically infected patients can block replications of these virus in the mouse model. It can protect virus derived from a genotype that's similar to the infected patients. And also on the right, you can see that the sera can delay or protect virus that is different than the chronically infected patients, suggesting that antibody can confer cross-genotype protections against XCV infections. So we further test the mechanism, what we think that our vaccine can induce broad cross-genotype protection. We want to understand the mechanism of it. Many work has been done to characterize these humoral response in XCV patients. So on the right is a cartoon representations of the E1, E2 proteins. We don't know the structures of E1 yet, and we have a partial structure of E2. The red highlighted the kind of conserved area that interacting with the host cell receptor CD81. And the number, these are highlighted many monoclonal antibody that has isolated from human patients. These antibodies are sufficient to confer cross-protective protections against many genotypes of antibody. So our question is whether that's highlighted that actually there's many effective neutralizing antibody can block infections of XCV, and most of these antibodies are blocking conserved functions of these glycoproteins. For example, the CD81 binding regions, many antibody are targeted to that. So basically, is that if the antibody can block conserved functions of the proteins, it can prevent many different genotypes to infect cells. So this experiment just highlight we use the vaccinated sera to see if we can block binding of various different monoclonal antibodies to E1, E2. The idea is that if the anti-sera can block binding of these monoclonal antibodies on E1, E2, suggesting that they might actually induce similar antibodies to those areas. And so this slide basically showed that on the red line, the vaccinated sera cannot block binding of various different monoclonal antibody onto E1, E2, but our vaccinated sera can successfully block binding of these various different monoclonal antibody binding on E1, E2, suggesting that we have antibodies targeted similar epitope to these effective monoclonal antibody. And because we're using E1, E2, we also see evidence that the vaccinated sera can also bind antibodies targeted to the E1 protein as well. And this is what we see. Similarly, the phase 1 clinical trial, the vaccinated sera can also block a variety of different monoclonal antibodies onto E1, E2, suggesting that our vaccine in human can also prevent induce antibodies similar to these effective monoclonal antibody. So one of the problem with HCV is it can easily mutate, sort of what you see in treatments, that they can acquire mutations that can escape from any control that you exert onto the virus. For example, we know that there's virus that can acquire mutations, can evade the antibody effects to block the infections. So we test, this experiment is to test whether our vaccine-induced sera can block any virus escape mutations. On the left is the control. As you can see, so in the y-axis, we're measuring virus infectivity. So more virus, you can see that the line is a little higher. And then if we can block infections, then you will see the line is lower. On the left is the control wild-type virus. We can test our GOAT, our vaccinated sera 757. It can effectively block infections of the wild-type virus. And so there's other three different monoclonal antibody, AP33, AR3A, AR5A, and AR3A. I'm actually really testing my eyesight here. And so, but on the middle and the right panel, basically we're engineering these mutations into the virus. Now you can see that the virus are resistant in the middle panel, for example, is now resistant to the neutralization exerted by AP33. But however, our GOAT sera can still maintain that neutralizations against this mutant virus. And the same thing also in the right-hand side, combinations of different variety of escape mutations do not block our ability of the vaccine-induced sera to neutralize the virus, suggesting that the virus can, our vaccine can induce multiple neutralizing antibodies to protect infections. So this is what we think has happened. The concept is very similar to monotherapy or multi-drug therapy. When you only have one neutralizing antibody or just a very similar class of neutralizing antibody, the virus are very easily to acquire mutation to escape from the control of the antibody and make it resistant to the neutralizations. But whereas in the vaccine scenario, you actually induce a polyclonal antibodies that target to multiple area on the envelope proteins or perhaps in the virus. In this case, even at one site that you have mutations, other neutralizing antibodies can actually block. And that's sort of what we think is happening in the previous slide. Even though you have selected resistant mutations, the vaccine-induced sera can actually still maintain that protections against the virus. So we're currently testing whether this is truly the case. When you actually prolong selections under the anti-sera, can we actually block mutations to arise in the virus? So this is sort of summarize some of all the data that I tell you. This E1, E2 glycoproteins based vaccine has been tested in humans. It's safe. It induce antibodies that bind to E1, E2 and actually confer protections in the chimpanzee data that I showed you on the earlier slide. And also it's induced cross-neutralizing antibody. And also there's evidence that this vaccine can induce T cells response, although I think there's a room for improvement. One thing that we didn't tell you is that the E1, E2, or perhaps I did tell you, is that the E1, E2 is very hard to make. So in order to kind of expand it into the clinic, we make a improvement from a bio engineering side of it, so we can help to make this in a much bigger scale so that we can actually bring it to clinic in a much more efficient manner. So on the left is a class, our previous way will basically express this E1, E2 in mammalian cells. We purify them from a lectin that bind the sugars on the E1, E2 to purify the proteins from the cells. On the left, we actually identify a insertion sites that we can actually add add a tag to the proteins, now we are able to have a handle to pull out these E1, E2 proteins in a much more efficient manner. The next few slides that I'm going to show you is a negative slide, but they are negative in a positive way. What I mean is that these slides basically highlight that in the left and the right, both the conventional method using GNA to purify the protein is similar to our weights using the FC tech to purify the protein. They basically able to different conformation-specific monoclonal antibody can pull down these proteins, suggesting that they fold similarly. And also, this is ELISA highlighting that these AR4A and AR5A is a conformation-specific antibody, and they are able to recognize either of these proteins similarly, suggesting that they maintain similar structure. And also, we tested in a small animal model, and either purification methods induce very similar protection effect to prevent XCV infections. And also the FC-derived E1, E2 can also induce cross-neutralizing antibodies to a variety of different genotypes of XCV. As earlier mentioned, besides neutralizing antibody, T cells is also critical for XCV protection. So we also, our strategy is to use conserved T cell epitope within the non-structural proteins of T cells, because our rationale is that using conserved epitope, we can actually target or direct the T cell response to conserved area of the XCV vaccine, and perhaps we can avoid some of the escape mutations that perhaps other T cell vaccine might have. And so we selected a few T cell epitope. These T cell epitope contain numerous CD4 and CD8 epitope, and they can also present it in HLA that is common to North America genotype. So we think that these will be a good strategies to induce T cell response in our vaccine. So this is sort of our vaccine is going to look like. We'll have a major component will be the E1, E2 recombinant proteins that's synthesized from CHO cells that's purified using the FC tech. And also we're going to have included a cocktail of XCV non-structural proteins to induce T cells response. Currently, we're actually manufacturing the vaccines in the University of Alberta under the GMP facility. So we are planning to do preclinical talks this summer. And if we are successful, this overcome a major hurdle, and we are hoping to bring this vaccine into the clinic in near future. So I would like to acknowledge our GMP team that's manufacturing these vaccines, and also Greg Corbett and Gail in the ACTM, because they are responsible for the GMP facility. Without them, we were never able to make the vaccines in U of A. There's also people that share their reagents and our funding support. And also, thank you for your attention. I'd like to also take the opportunity to acknowledge Mike and Lawrence for their support and leadership. Without them, this program will not get to where we are today. So thank you.